AMALearning, you're about to. Specification 0.3.10b. Describe how genetic variants in the coding DNA of a gene can affect phenotype by altering the sequence of amino acids and therefore the activity of the protein produced. To successfully answer any GCSE questions linked to this topic, you will need to be able to describe how a genetic variant or mutation within the coding part of the DNA will impact on the phenotype. What is coding DNA? Coding DNA is the part of DNA that acts like a recipe to make proteins. It provides the exact instructions for the sequence of amino acids, which are the building blocks of proteins. These proteins then carry out essential functions in the body. What happens when coding DNA changes? When the sequence of coding DNA changes, a mutation occurs, it's like altering a recipe. If the mutation changes the protein shape, it might not work properly. For example, an enzyme may no longer fit with its substrate, like a broken key that doesn't fit the lock. This can cause diseases or disorders. Additionally, the mutation might lead to the production of alleles, different versions of the same gene. Some versions of the gene may help an organism survive. What are the real-life examples of this? If an enzyme's shape is altered, it might not perform its job in chemical reactions, leading to problems like metabolic disorders. If the food substrate does not fit into the digestive enzyme, then the food will not be broken down. In addition to enzymes, collagen and similar proteins are known as structural proteins. They give strength to skin and tissues. Mutations or variants in the coding part of the DNA might cause the structural proteins to become weaker and cause conditions like brittle bones or joint issues. How common are these changes? Most mutations don't cause noticeable changes and are harmless. Some mutations can be harmful, leading to diseases, while others might be beneficial and give advantages, such as better survival in certain environments. Genetic variants can happen in both coding and non-coding DNA. Some have no impact, while others can drastically affect the way proteins work, influencing traits and health. Let's apply what we have learnt to a past paper question. In this question, we are being asked to explain why alleles can produce proteins with different structures. This means we must use this lesson to describe why alleles are produced and answer the remainder of the question using specification 0.3.8b with a specific focus on transcription. We know from our learning that when the sequence of coding DNA changes, a mutation occurs. It's like altering a recipe. If the mutation changes the protein shape, it might lead to the production of alleles, different versions of the same gene. We also know from 3.8b that the mRNA, messenger RNA, attaches to a ribosome. The ribosome reads the mRNA instructions in triplets of bases called codons, which tell it which amino acids to use. tRNA, transfer RNA molecules, bring the right amino acids to the ribosome based on the triplets of bases, or codons. The ribosome links these amino acids together in the exact order specified by the mRNA, forming a polypeptide chain, a long chain of amino acids. If we compare our learning to the mark scheme, we would have got the marks outlined in every single bullet point, so full marks. Well done. Our animations were designed by experienced UK-trained teachers. We have been achieving amongst the best results in the country for many years, and we decided it was time to share our success. Have you just AMALed and want to AMAL some more? Don't forget to like, subscribe and share to keep yourself up to date with the latest uploads. AMALearning, you just did.